My name is Phat with the USDA-ARS National Peanut Research Laboratory in collaboration with Auburn University. We have studied leaf spot resistance in peanuts, and my talk today is going to be on the identification of disease-related genes associated with leaf spot resistance in cultivated peanut and their conversion to gene expression marker. Early leaf spot, or ELS, caused by Cercospora arachnoidicola, and late leaf spot caused by Cercosporidium personatum are serious foliar diseases in peanut. Its typical symptoms of ELS are brown lesions surrounded by yellow color on the upper leaf surface, and the symptoms for late leaf spot exhibit dark brown or black lesions on the underside of the affected leaves. The fungicide treatments can reduce the severity of leaf spot diseases. However, it can increase costs as well as environmental pollution and incidence of other diseases. Therefore, development of peanut varieties with leaf spot resistance is an effective and sustainable strategy to manage foliar diseases. Plants are naturally exposed to different environment and different pathogens. So it has suberin or waxy particle on the structure surface, as well as the cell wall and hairs to protect it. But then once the pathogen penetrates the cell, then it's recognized by pattern recognition receptors, or PRRs, representative of receptor-like kinases or receptor-like proteins that can activate a pattern trigger immunity response. Non-adaptive pathogens are stopped at this point because the cells can activate calcium release as well as reactive oxygen and also phytohormones and activation of transcriptional factors. The adaptive pathogens can penetrate the cells at this point to release pathogenic effectors and activate the second line of defense, the R proteins or resistant proteins that can stop the pathogen called effector trigger immunity, or ETI. The combination of PTI and ETI worked effectively with different pathogens. However, new pathogens with effective or better ways to penetrate the plants can overcome the defense mechanism. So in resistant gene identification, the physical structure or the motifs are very conserved, so it's been cloned in many different plant species. In peanuts, more than 400 different potential resistant gene candidates have been identified, with 214 were actively expressed in leaves in our study, and 89 of those resistant genes or genes were analyzed for differential gene expression. Recent research on leaf spot resistance has identified major and minor QTLs, indicating that the leaf spot resistant trait is complex. Zhang et al. In 2020 identified two major QTLs for both ELS and LLS on chromosome B9, as well as 4 QTLs for ELS and 40 minor QTLs through GWAS analysis utilizing the US Minicore collection. Chu et al. In 2019 identified 3 QTLs for ELS, 2 on chromosome A3, 1 on B3, and 3 QTLs on B3, A5, and B5 for late leaf spot through QTL genomic mapping utilizing the recombinant inbred lines from Florida 7 and GPNC-WS16 cross. Han et al. In 2018 identified 1 QTL on chromosome B5 for late leaf spot and 2 QTLs on A3 and B4 for early leaf spot through genotyping by sequencing, or GBS, and QTL mapping. Our objective is first to evaluate peanut breeding lines for leaf spot resistance in the field, then to generate real-time PCRs of the R gene expression using RNA from susceptible and resistant lines, then to compare the gene expression profiles of these R genes to leaf spot resistance and to convert to DNA molecular markers to be applied in breeding programs. 48 different lines were grown, including 45 breeding lines, and 3 chicks were planted in randomized complete block design with 4 replicates at the Herc Center in Dawson, Georgia in 2017. Fungicide was withheld to induce diseases, and other treatments are according to the best management practice. Visual assessment of leaf spot disease was progressed and was made according to Florida's 1 to 10 scoring system at around 130 days after planting. This is an area view of the disease plots at the Herc Center in Dawson, Georgia in 2017. These are about 20-foot twin rows comparing to some more susceptible lines versus some of the tolerant lines. This graph shows the distribution of leaf spots disease severity among the peanut genotypes in 2017. Many of them are clustered in the disease rating of less than 5, and the checks are also included for reference. For gene expression experiments, we decided to use 4 of the susceptible lines, and for the resistant lines, and 3 checks. You can see that these are 17 identifiers with its appropriate causes and its relative ratings. In terms of tissue collection and RNA analysis, 4 leaf samples were collected per row treatment, and there are 3 row replications per tested genotypes. RNAs were extracted from each leaf sample separately, pooled for each individual row samples, and the representative pool samples were utilized for gene expression study. Real-time PCR was performed on a QuantStudio 7 using a comparative CT method, the delta-delta CT. The gene expression level or relative quantification is the negative delta-delta CT log of 2. The relative quantification values of peanut genotypes were graphed together for a relative comparison of the gene expression levels. This graph shows a relative quantification of gene expression levels with the susceptible lines in red and the resistant lines in green, with the checks in the gray bars. The x-axis represents the different genotypes, and the y-axis represents the different levels of relative quantification. This number represents the average of the susceptible lines. This number represents the average of the resistant lines. Correlation with the susceptible versus resistance is a negative correlation of all these 6 genes represented here. As you notice, Tifgard and Giorgio 6G usually clusters with the susceptible lines, where the AU-NPRL-17 usually clusters with the resistant lines. Again, this graph shows a relative quantification of the susceptible versus resistant lines here, maintaining a negative correlation for all these 6 genes shown, with the most susceptible line consistently being AU-1708 and the most resistant line consistently being AU-1741, close to AU-NPRL-17. This table shows a composite of 36 different R genes that have similar patterns as the last 2 slides, where the genes are inclusive in this list, where the target ID is shown in this column. The fold differences in terms of negative correlation between susceptible versus resistance, the chromosome number with the color blue highlighted for more than 1 chromosome location number, the gene function identification based on the BLAST-X results, and the number of SNPs based on the cloning and sequencing of these different particular size products, and with the yellow being more than 3 SNPs per product. This shows the results of the 2018 planting compared to the 2017. We can see that the ones that we selected for 17 are on this list as well for the most tolerant lines. And we will use these information to extract genomic DNA and use this for genotyping in the future for the 2018 as well as future plantings. This slide demonstrates how this information could be used to develop molecular markers where we clone and sequence a serine threonine protein kinase, PCRK1, from susceptible versus resistant. What we find is susceptible has an AB allele, where the resistant has a duplication of A allele, homozygous, and we can see the SNPs at different locations. We can identify a indel as well, where we can produce a PCR product and generate different sizes for genotyping application. There are several SNP genotyping assays available. The earliest was TACMAN, and recently CASP type of marker, and now there's an RH-AMP genotyping system as well using RNase-H. But they all require allele-specific primer, 1 and 2, separates the different alleles, some sort of probe, and then Taq polymerase in PCR reaction, separation on a analyzer and data analysis to verify homozygous versus heterozygous. In our ongoing and future research, we plan to extract genomic DNA from lines that shows resistant versus susceptible to leaf spots, and to apply these Snip markers to these lines, then to correlate the homozygosity or heterozygosity of these markers and the gene expression patterns of the R genes to leaf spot resistance for verification. Then in the long term, develop new breeding populations to establish high yielding lines with leaf spot resistance and apply these novel genetic markers to facilitate selection. Thank you.